adaptive immunity and where white blood cells are produced. So please know that the bone marrow stem cells is what makes platelets, blood cells, and white blood cells. Please make sure you watch this video. To remind us, adaptive immunity is a specific response to a specific pathogen. Our lymphatic system is involved in adaptive immunity. Specifically, the T cells are developed in our thymus from our bone marrow, and they are an important player for our immune system. Adaptive response in our immune system relies on lymphocytes. Lymphocytes are white blood cells. They are produced by stem cells in our bone marrow, and there's two types of lymphocytes that are responsible in our immune system for identifying pathogens. The first one is T cells. These mature in the thymus, as I previously stated, and there's two kinds to be familiar with, helper T cells and cytotoxic T cells. The other type are B cells. B cells stay and mature in bone marrow. B cells give rise to plasma cells, thankfully, because plasma cells produce antibodies, which I will get into a bit on the next slide. Antigens are any substance that elicits a lymphocyte response from our immune system. Looking at the picture, you can see there's antigens on the outside of that pathogen. So this could be, the pathogen again could be a bacterial cell or a virus. And the antigens are molecular markers on the outside of that cell. Typically, antigens they can be proteins, they can be polysaccharides, long chains, again, of sugars. If you look very closely at the diagram, you will see that the antigen is being circled, and the antigen can be quite a large molecule, but only part of that molecule is going to bind to an antigen receptor on the outside of the B cell. So when we're talking about the immune system, something that I haven't really mentioned is it's all about cellular recognition, and how cells identify themselves are from the outside, and those molecular markers, those antigens on the outside, and whether or not they bind or not. And if they don't bind, that could red flag our immune system that there's something, a cell or an intruder or some substance that doesn't necessarily belong. So back to this picture, so you can see the antigen is a large molecule again on the outside of that pathogen. The part that the B cell directly binds to, like a key in a lock, is called the epitope. From that key in the lock, that's going to trigger the B cell to be activated, essentially, and for antibodies to be made, and I'll talk more about antibodies on the next slide. So what is an antibody? Antibodies have another name. When they're large, they're known as immunoglobulin, and these are proteins that are made by B cells that are going to bind to the antigens. So again, the B cell needs to be activated by the key in the lock, by that receptor matching up with the epitope, and when that matches, antibodies are going to be produced. Antibodies usually have a Y shape, so if you look at the picture on the slide right now, it's upside down, but it has like a Y shape to it. These match the antigen receptors. They need to match, and they're secreted. They're not attached to the membrane anymore of that B cell. They're secreted so that way they stick to those pathogens, which is going to be very important in disabling pathogens from doing harm to our body. There are millions of different B and T cells produced by the immune system. Now, what makes them different from one another are that on the exterior or the outside of those cells are specific antigen receptors. Now, this is something I was saying in class the other day. Please note that whenever the book illustrates a cell like a B cell and shows you the antigen receptor on the outside, it's very deceiving because you think that that one cell is exclusive with that one shape and it's going to be exclusive with one type of pathogen. So sometimes in books it's a little bit deceiving, the diagrams, but an actual cell can have 100,000 different kinds of receptors on its membrane. So although in the books it shows us one or a couple different kinds, there's hundreds of thousands of different kinds on the outside of one cell. So please make sure that you understand that these cells, there's millions of different kinds with millions of different kinds of combinations that can be on the outside, meaning the antigen receptors on the outside. Now, you don't have to know much about this slide, but it definitely is a really good review of transcription and translation and how when we look at a strand of DNA, how only parts of it are useful, at least for making this polypeptide antigen receptor on the outside of a B cell. So I would definitely just kind of glance over it, make sure you understand step 1 down to step 4. In terms of how proteins are made, and antigen receptors being with this chapter. The origin of self-tolerance in how cells like our lymphocytes don't attack one another. The reason is with the last slide showing you transcription, translation, how the receptors are made, because again, that's the key for identification. Undifferentiated B cells, there is an immunoglobulin gene, an antibody gene, where again the DNA gets rearranged, there's alternative splicing taking place, which is going to lead to many different kinds of antigen receptors on the outside. I wasn't very specific, I think a slide or two ago, but there's a million different B cell antigen receptors and 10 million different T cell antigen receptors that are made from this one gene. So again, it always goes down to the rearrangement and the splicing. So as lymphocytes mature in bone marrow or in the thymus for the T cells, they are tested out for self-reactivity. And you'll see a picture on the next slide that I think summarizes this really well. B and T cells with self-receptors are programmed for cell death, or they are labeled as non-functional and they will eventually be dead. Another good diagram of how from stem cells, there are different kinds of B cells. Again, these B cells only show you one antigen receptor per cell, which isn't realistic, but it's a very simplistic way of looking at these antigen receptors. So again, please note, it could be hundreds of thousands of those things on the outside. But looking at the 5 different kinds of B cells that came from that stem cell, 2 of them are eliminated right out the gate. Again, for whatever reason, they will attack each other and based on their antigen receptors, so they have to be programmed for cell death. Then there's antigens out there that's able to bind to that antigen receptor. The B cell is triggered and plasma cells are able to be formed and to make antibodies, and those antibodies are going to bind to the antigens on the pathogen. So this is a simple way at looking at how B cells are made and how that comes into play with pathogens.